Welcome back to the Worm Podcast. It is just incredibly good to have you with us again. We are diving into some truly mind-bending biology today. But before we get into the molecular weeds, I actually have a quick favor to ask you. If you are enjoying these deep dives, please take just a second to follow, like, comment, and, share the show with your colleagues and friends.
Yeah, it really does make a massive difference in helping the community thrive.
Exactly. We are on a serious mission to grow this nematode-focused community, and your support is basically the engine that allows us to keep bringing you these stories from the frontiers of microscopic biology. So, let's get into the main event. Today's deep dive is centered on a brilliant 2026 PLOS biology paper by Chang and colleagues.
Right, a really fascinating piece of work.
It really is. And since you are already an active part of the Worm podcast community, we are going to completely skip the basic introductions to C. elegans. You already know why these incredible little nematodes are the gold standard for studying aging and genetics, so we're just jumping straight into the deep end.
I love that. No need to rehash the basics.
Exactly. Our mission today is to explore the invisible molecular landscape of something called heat hormesis. Essentially, we are looking at how a transient dose of, mild heat stress gives our favorite worms a literal survival superpower.
Yeah, protecting them from lethal temperatures and extending their overall lifespan.
Right. So, set the stage for us. Why is this such a breakthrough?
Well, to see why this is a breakthrough, we kind of have to look at the broader biological context. The concept of hormesis. The idea that a small, manageable dose of stress triggers protective mechanisms that are broadly beneficial is ubiquitous.
Like what we see in humans, right?
Exactly. We see it in yeast, we see it in fruit flies, and yeah, we see it in humans when we subject our bodies to a hot sauna or an ice bath. The organism detects a mild threat and basically fortifies its defenses.
But the mystery has always been how it actually works under the hood.
Precisely. What hidden molecular switches are being flipped to grant that resilience? What biological trade-offs does an organism have to make internally to survive a harsher environment? The Chang paper is a milestone because it finally isolates the specific levers being pulled inside the genome to make that long-term stress resistance a reality.
Okay, let's unpack this experimental regimen. Because before we can even begin to understand the molecular magic happening in the genome, we really need to observe the physical survival superpower this heat priming actually gives the worms.
Right, the physical phenotype is where it starts.
So the researchers designed a very deliberate timeline. They start with young adult worms living happily at a comfortable standard 20 degrees Celsius. Then they initiate what they call the priming phase. They dial the temperature up to 30 degrees Celsius for 6 hours.
And for a nematode, 30 degrees is distinctly uncomfortable.
Right, it's not lethal, but it's a mild stressor.
Exactly. That 30-degree mark is critical. It pushes the worm's physiology out of homeostasis, which forces it to mount a defensive response without actually causing catastrophic tissue damage.
Makes sense. Then, after that 6-hour priming, they give the worms a 12-hour recovery period. They drop the temperature back to that cozy 20 degrees. And finally, after they've recovered, the researchers hit them with a severe, potentially lethal heat shock. We're talking 35 degrees or 37 degrees Celsius.
Which is a massive spike for them.
Huge. When I visualize this setup, it feels like giving the worms a stress vaccine. We are giving them a highly targeted, controlled sauna session to prepare their cellular defenses for a catastrophic heat wave that might strike a day or two later.
The stress vaccine analogy perfectly captures the biological intent there. And the physical phenotype, like the actual observable outcome of administering that vaccine, is dramatic.
What exactly happens when the heat wave hits?
Well, when the lethal 35-degree heat shock hits, the naive worms, the ones that stayed at the comfortable 20-degree baseline the entire time, they just collapse. Their survival rates plummet almost immediately.
Oh, wow. Just immediate collapse.
Yeah, they are completely paralyzed. But the primed worms, the ones that received the mild stress earlier, survive significantly longer. Furthermore, they maintain far better motility after enduring a severe 6-hour heat shock. What's fascinating here is the concept of memory.
I want to dig into that concept of memory, actually, because that implies the worm holds on to this information.
They really do. The researchers demonstrated that the worms retain the protective benefits of that initial mild stress long after the threat has passed.
Even after they've cooled down.
Exactly. They maintain this enhanced survival superpower after 12 hours of recovery in comfortable temperatures, after 48 hours, and remarkably, sometimes even up to 96 hours post-priming. They're basically holding a biological grudge against the heat.
Hold on, I need to push back on this logically. Because if I'm a worm and I've been chilling in a perfectly comfortable 20-degree environment for 12, 48, or even 96 hours, shouldn't my biology have just forgotten the mild stress by then?
You would think so, yeah.
Yeah. Right, because the environmental threat is clearly over. The coast is clear. And 96 hours in nematode time is a massive percentage of their lifespan. Shouldn't all those cellular alarm systems have powered down to conserve energy?
It's a great question.
I mean, where exactly is this memory being stored if they're just swimming around normally in a safe environment for days?
The logic of your question gets to the very heart of the mystery. From a macroscopic perspective, just observing the worms swimming in a dish at 20 degrees, everything looks completely normal. Their behavior has totally returned to baseline.
Right, so the secret has to be microscopic.
Exactly. To answer where that memory is physically stored, the research team had to transition from looking at macroscopic survival traits to mapping out the invisible molecular landscape inside the nucleus.
So they had to basically crack open the genetics.
They did. They utilized a multi-omic approach, which gives us a sort of two-dimensional view of genetic activity.
Hmm.
First, they used RNA-seq, which captures the transcriptomic data.
Which tells us exactly which genes are actively being transcribed into RNA to build proteins, right?
Exactly. And second, they paired that with ATAC-seq. ATAC-seq basically maps chromatin accessibility.
Just to make sure we are all on the same page here, chromatin accessibility refers to how tightly coiled or loosely packed the DNA strands are around their structural proteins.
That is the core mechanism, yes. If the chromatin is open and accessible, the molecular machinery can get in there and turn those genes on. If the chromatin is wound tight and closed, those genes are locked away in the vault.
Makes sense.
So immediately after that 6-hour mild priming at 30 degrees, the data shows exactly what we would expect. Thousands of genes dramatically turn on or off, and massive shifts occur in how open or closed the chromatin is across the genome. The worm is actively fighting the heat.
But then there's a twist, right?
A huge twist.
Here's where it gets really interesting, because the researchers looked at the multi-omic data after the 12-hour recovery period, and practically everything vanished.
It just disappeared.
It's like watching a construction crew open a master blueprint, build a massive, complex scaffolding system all over a building site to deal with an incoming hurricane. But the absolute second the wind dies down, they instantly tear all the scaffolding down and lock the blueprint back in the vault.
That is a perfect way to visualize it. The RNA-seq and ATAC-seq data show that the genome reverts almost completely to its baseline state.
But if the genes turn off and the chromatin closes back up, how does the worm remember the storm when the severe heat wave hits 48 hours later?
The punchline of this multi-omic disappearing act is that the molecular memory remains completely invisible until the second severe stress hits.
So they don't see it coming until it's there.
Right. When the catastrophic 35-degree heat shock occurs, the primed worms do not just react faster or stronger. They mount a completely different, highly specialized molecular defense that the naive worms simply do not possess.
They aren't just replaying the greatest hits from the first stress response.
Far from it. When naive worms face the severe heat, their molecular response is chaotic. But the primed worms deploy a strategic defense. For example, they uniquely downregulate massive swaths of their lipid metabolism pathways.
Wait, why shut down lipid and fat metabolism during a heat wave?
Well, consider what happens to butter when you leave it on a hot stove.
Right, it melts.
Exactly. Cellular membranes are built out of lipids. When environmental temperatures skyrocket, those lipid membranes become dangerously fluid. They lose their structural integrity, and the cells essentially risk turning to soup.
Oh, wow. Turning to soup sounds bad.
It's very bad. So by actively downregulating specific lipid metabolic pathways, the primed worms alter the composition of their membranes. This compensates for that heat-induced fluidity and keeps their cells intact.
That's incredibly smart.
It is. Furthermore, they fundamentally alter their collagen expression to reinforce their physical structure, and they activate unique stress response networks. The initial mild stress left a latent epigenetic imprint, a hidden biological tripwire that only triggers when a lethal threat appears.
That is astonishing. The memory is literally a hidden tripwire waiting for the temperature threshold to break.
Exactly.
But this molecular imprint does not just protect them from severe heat. The paper explores how this actually alters their fundamental aging process. However, the researchers discovered that acquiring this longevity requires a very surprising biological sacrifice.
The biological cost is profound. To uncover it, the researchers set up a brilliant comparative model. They contrasted wild-type worms, your standard, healthy C. elegans, with a specific mutant strain known as GLP-1ts.
For those tracking the genetics, the GLP-1T's mutants are fascinating because at specific temperatures, they actually fail to develop a germline. They effectively lack a reproductive system.
Right, and the absence of a germline makes the GLP-1T's mutants naturally long-lived compared to wild-type worms.
Okay, so they already have an advantage.
They do. Now, the researchers applied the heat hormesis regimen to both groups. They found that the mild heat priming significantly extended the natural overall lifespan of the wild-type worms. However, it provided absolutely no lifespan extension to the GLP-1 mutants.
Which completely isolates the mechanism. The lifespan extension in normal, healthy worms is directly dependent on their reproductive system.
Exactly. The data points directly to the reproductive tissues. When the researchers investigated the wild-type worms during that crucial 12-hour recovery window after the mild stress, they observed a temporary but severe disruption in reproduction.
What did that disruption look like?
The worms exhibited a heavily reduced brood size, and they laid a significant number of inviable eggs.
So what does this all mean? If I can bring back my industrial analogy from earlier, it's like the worms are deliberately hitting the pause button on their factory production line, which is reproduction, so they can rapidly divert all of the factory's electricity, all its raw materials, toward fortifying the walls of the factory itself to weather the storm.
The factory analogy captures the resource allocation perfectly. Somatic maintenance, things like repairing tissues and producing protective proteins, requires a tremendous amount of cellular energy.
And reproduction is a huge energy drain, too.
Reproduction is arguably the most energy-intensive process an organism undertakes. When the mild stress registers, the wild-type worm performs a brutal biological calculus. It intentionally sacrifices a small window of reproductive output to ensure the organism itself does not die.
That is wild.
And if we connect this to the bigger picture, this transient germline pause yields massive long-term dividends for the organism's health span.
How do those long-term dividends manifest as the worm ages? I mean, what's the lasting benefit?
Under normal conditions, as a wild-type worm enters early adulthood, its biological priority shifts entirely to reproduction. Consequently, it rapidly loses the ability to mount a robust heat shock protein response.
Right, its natural cellular defense system degrades because the energy is being poured into generating offspring.
Exactly. However, by forcing that temporary reproductive pause via mild heat priming, the wild-type worms entirely preserve their ability to induce massive amounts of heat shock proteins much later into early adulthood.
That is an incredible trade-off. A temporary sacrifice in reproductive output yields a permanent structural upgrade to healthy aging. They basically retain their biological youth, their capacity to fight off stress, simply because they strategically paused the system.
You nailed it. The germline and the somatic cells are locked in constant communication, basically constantly negotiating over a finite pool of resources.
And the heat stress tips the scales.
Exactly. The mild heat stress forcefully changes the terms of that negotiation, shifting the balance of power toward long-term survival and maintenance over immediate reproduction.
Okay, we now know what is happening under the hood. We have chromatin accessibility shifts, hidden epigenetic tripwires, and strategic germline pauses. And we clearly know why it matters, given that it extends lifespan and grants severe heat resistance. But who is actually pulling the genetic levers to make this highly coordinated defense a reality? The genome doesn't just spontaneously restructure itself.
No, it doesn't. It requires a highly sophisticated management team of molecular regulators.
And I am thrilled to highlight the incredible detective work the researchers did here. They used motif analysis, looking at the exact sequences of DNA that were opening up during the stress response, searching for the specific biological keys that fit those locks.
It's a really elegant approach.
Then they systematically used RNAi knockdowns to eliminate these suspects one by one, watching to see which removal broke the stress memory. And they uncovered a specific set of master managers, HSF1, ELT2, DPY27, and MARS1.
The identification of these regulators is crucial because they are highly conserved evolutionarily. Homologs for these genes exist across the animal kingdom, including in human biology.
Which is why we care, right?
Exactly. We always anticipated HSF1 would be involved. It is the classic master regulator of the heat shock response. But the other managers revealed the true complexity of this hidden memory. ELT2, for instance, functions specifically in the intestine.
Wait, why would the worm's intestinal tract be acting as a manager during a whole-body heat wave?
Because the intestine in C. elegans acts as the primary metabolic hub. It is the tissue that manages the lipid storage and synthesis we discussed earlier.
The butter melting problem.
Exactly. The requirement of ELT2 demonstrates cross-tissue communication. The intestine is actively sensing the stress and coordinating the metabolic shift for the entire organism.
That makes a lot of sense. What about the others?
Then we look at DPY27, which is a protein responsible for managing the actual 3D architecture of the chromosome. It physically condenses and structures the DNA loops.
Which provides the physical mechanism for the chromatin accessibility changes observed in the ATAC-seq data, right?
Precisely.
And how does Mars-1 fit into this management team?
Mars-1 operates at the translation level. It is responsible for attaching the amino acid methionine to transfer RNA during the synthesis of new proteins. Its involvement proves that regulated translation, literally controlling how the protective proteins are actually manufactured on the ribosome, is just as vital as the initial transcription of the genes.
Okay, but among all the regulators the research team identified, the one that truly stands out as the architect of this stress memory is FOS1.
Absolutely. FOS1, which is a critical component of the AP1 complex, operates as what molecular biologists term a pioneer transcription factor.
A pioneer factor. That implies it is venturing into uncharted territory on the genome.
The distinction is vital. The vast majority of transcription factors are opportunistic. They can only bind to their target DNA sequence if the chromatin is already loose, open, and easily accessible.
So they need the door to be open for them.
Right, but pioneer factors like FOS1 possess the unique biochemical ability to target closed, tightly packed, inaccessible chromatin and physically pry it open.
Wow. So FOS1 is the molecular brute force that actually writes that invisible epigenetic memory. It wedges the condensed DNA open just enough to plant that latent tripwire, ensuring the protective genes are primed for the next severe heat wave.
It creates that persistent epigenetic footprint. Even when the majority of the genome returns to baseline during the 12-hour recovery, the pioneer activity of FOS1 leaves a permanent structural mark that allows the worm to mount its massive specialized defense days later.
This leads me to a fascinating theoretical question. If specific transcription factors like the pioneer FOS1 and the master regulator HSF1 are the literal biochemical keys unlocking these massive anti-aging and stress resistance benefits, could we theoretically just give these genes a nudge artificially?
You mean without the heat?
Yeah. Could a therapeutic intervention simply turn on FOS1, completely bypassing the need to subject the organism to physiological heat stress at all? Could we synthesize the benefits of the sauna without ever having to sweat?
The prospect of pharmacological intervention is alluring, I'll give you that, but it brings up a fundamental rule of biological networks. On paper, identifying a molecular switch suggests you can flip it synthetically, but transcription factors do not operate in a vacuum.
They need the rest of the team.
Exactly. FOS1 is part of a massive, dynamic multi-protein complex. It requires the context of ELT2 signaling from the intestine. It requires the germline to pause reproduction. It requires DPY27 to restructure the chromosomal loops.
It is an intricate biological symphony, not a solo performance.
That's a great way to put it. If you artificially force the FOS1 switch into the on position without the broader physiological context of mild heat stress, you risk disrupting a very delicate equilibrium.
What's the worst that could happen?
Well, you might induce the heat shock protein, sure. But because FOS1 and the AP1 complex heavily regulate cell growth and development, you might also permanently sterilize the organism, or worse, trigger runaway cellular proliferation, leading to tumors.
So definitely not a solo performance you want to mess with.
No, the mild physical stress acts as the maestro of the symphony, ensuring that the timing, the dosage, and the tissue-specific crosstalk are executed perfectly.
You cannot just hack a single node in the network and expect the entire organism to play the correct tune. The physiological context of the stress is the necessary trigger.
Exactly. To synthesize the extraordinary journey this research provides, a transient mild heat stress activates highly conserved regulators, notably the pioneer factor FOS1, to physically remodel the chromatin architecture.
Right.
This genetic cascade forces a strategic temporary halt in reproductive outlet. That brief reproductive sacrifice pays massive biological dividends, granting the organism long-term physical resilience, preserving its heat shock defenses deep into adulthood, and ultimately extending its lifespan.
It is a breathtakingly elegant system of survival, and it leaves you with a deeply provocative thought to explore on your own. We know that this epigenetic memory alters the trajectory of the worm that experiences the heat. But think about human biology for a second.
Oh, right, taking it back to humans.
Exactly. The study mentions that in humans, sauna therapy is linked to reduced cardiovascular mortality and improved health span. If mammalian AP1 complexes act similarly to the worm's FOS1, reshaping chromatin in response to mild stress, what invisible biological trade-offs is your own body making when you sit in a hot sauna or take a cold plunge?
That is a fascinating question.
Is your body temporarily pausing other cellular functions to write a memory of youth into your DNA? It fundamentally changes how we think about the legacy of environmental stress and whether our own passing encounters with adversity might be writing invisible armor into our genetic code.
The potential for transgenerational or even just long-term epigenetic impacts in this context is a thrilling frontier for future research.
It truly is. Well, before we sign off, I have an important announcement. If you are listening and you would like your own research or a paper you are passionate about discussed in a future deep dive on the podcast, please reach out and contact Dr. Viren Chauhan.
Yes, please do. We're always looking for groundbreaking data to unpack, and we would love to feature the work driving your field forward.
We definitely look forward to seeing what you send our way. Thank you so much for joining us on this deep dive. Keep asking questions, keep exploring the microscopic world, and we will catch you on the next one.